Thank you very much, Tim and Nancy and everyone else for hosting this. I'm really excited to get started and show everyone some different stuff for this next tutorial session. We're going to be looking at using R in hydrology, and I will be going over first an example of R in hydrology as well as a hands-on demonstration. So I think with that, I can go ahead and get started. I'll go ahead and start sharing my screen.
Yep, please go ahead.
Okay, great. And so just so everyone's aware, I'm also going to be posting, let's see, some links in the chat box while I go through some of these slides. So that may just take me a moment to switch back and forth between the two.
And I'll translate those links over to Discord as well. So Michelle, when you stick them in, I'll copy them into the Discord site too.
Okay, great, thank you. Okay, so I'm sharing my screen and hopefully the Zoom chat box is not covering it. Hopefully it's just the R slide. Okay, so thank you everyone. My name is Michelle Newcomer and I am a research scientist at Lawrence Berkeley National Laboratory. And today I'm going to be talking about using R in hydrology. And as I mentioned before, I'm going to show a few examples of doing this. One being an example at the large continental scale. And then another example, which we'll do together as part of this tutorial, using a Jupyter notebook in a web browser. And that will be an example from the Russian River watershed, where I have done much work over the past few years characterizing the impacts of fires on that watershed. And so you'll see how an analysis in R could be done using these type of tools. So before we begin, for the hands-on part of the tutorial session today, we will be using the Firefox web browser. So if you don't have Firefox, now would be a good time to download that and get that going. Unfortunately, sometimes one of the R packages in the Jupyter notebook, it doesn't work as well in Chrome. So Firefox, it works fine. So I ask that everyone have Firefox installed for that today. So I will go ahead and move on. So this workshop began, well, I started developing this workshop in 2018 when I was invited to give a workshop on big data. And specifically, this is part of the Lawrence Berkeley National Laboratory California Water Big Data Challenge. And so that's when I started developing these type of tools using Jupyter notebooks and binders so that people could run the notebooks online without having to install R or Python or any other software. So that was, I'll post these links as well in just a minute. I further developed this workshop when I was asked to give this workshop again at the International Association of Hydrologic Sciences conference that was held last year. And this is part of the Young Hydrologic Society. And you can find a lot of resources on what this Hydrologic Society is about and what we're doing. So during that workshop, we had other guest speakers, and they talked a lot about some of the new tools that are emerging using R, such as kriging approaches, which that question came up earlier about how can you include like geostatistical heterogeneity. Well, you can use these type of approaches. And there's also a new paper by one of our colleagues here that I will talk a little bit about today in Hydrological Earth System Sciences discussion. And then finally, I continued developing this workshop as part of the AGU Department of Energy Early Career workshop, and that's the one I gave in December of last year, where Tim and others got to be a part of that and see what we were up to. So I'm going to go ahead and post some of these links now in the chat so you can take a look at those resources. So the short course is really geared for any researcher who is interested in learning about applying R to a specific hydrological application. And this workshop today, I'm focusing on some simple tools to fetch data sets and do a simple analysis. But there's many more tools out there, including hydrological models, numerical methods, and other packages that are developed by the user community. And so it's a very well-supported community of hydrologists who are interested in using R for their applications. Oh, and as I mentioned before, if you're just joining us now, please have the Firefox web browser installed. That will be part of our hands-on session a little bit later today. This workshop has also been inspired and developed by many other people. I'm not the first one to put something like this together, and certainly all the tools I'm showing you today, they've been shown by other people as well. So there's a very large community of people involved in this type of research and this type of workshop development. So I'd like to give just credit to all those people who are doing this. Specifically, there's been workshops at EGU using R in hydrology, and there are links provided, which I will put in the chat window. There's links provided to those workshops. There's other presentations and tutorials on their Hydrological Society GitHub page. And then there's this paper that came out last year by Slater et al., really looking at how hydrology is being used today, including recent developments and future directions. So I went ahead and put them in the chat window right now, some of those links. And R is growing. There's been a significant advancement in the user community as well as the development of hydrological packages. And so here, the CRAN task view, this is essentially a web page showing all of the different packages specifically for hydrology. And this is developed by Zipper et al. More than 100 packages as of June 2020. This is driven by the increase in publicly accessible data as well as increases in computational power, analysis of complex data sets, such as the application that I'm going to show you at the CONUS scale, continental United States scale. And there's an active and supportive community of users. Also, people are using this for a lot of types of applications, including data acquisition, analysis, modeling, statistics. It's also used as a GIS, so you can visualize your data and show it in map form. We're going to be looking at a GIS application today. So you'll see an example of how you can use this for looking at water quality parameters in California. And you can also use it to publish. So for example, using our Markdown, you can have your model and your code all built into your paper and then publish that as your paper with your associated data sets with your paper so that everything is reproducible for your analysis. So let me also share some of those links with you.
So Michelle,
I know that there's a lot, yes.
When you when you post into the chat window, could you be sure that you're going to all panelists and attendees so that everyone sees the links? It may be only going to the panelist as a default.
Oh, I'm so sorry. Yes, you're right. It said by default, just all panelists. I'm sorry, everyone.
So I think Tim has caught up on some of the earlier ones, but just moving forward, it'd be easier. So.
Okay, yes. And also, do I need to repost some of those links?
I think Tim caught the earlier ones, so I think we're good. So just.
Okay.
Yep.
Okay, great. Many apologies about that. I didn't catch that. So thank you, Nancy, for pointing that out. All right, let's see. I think I posted this one. This is slide 5, and this is the CRAN link. Okay, perfect. Okay, so for today's workshop, as I mentioned, there's going to be two applications. The first is going to be a demonstration, and by demonstration I mean an example of some research that I've done at the continental scale using some of these tools. And obviously I can't do that with you here because I have to use NERSC for that specific application because there's so much data and computational needs associated with that. But the other one will be hands-on, and that one we'll all do together. And also after the workshop, you can watch the video that I did in 2018 on this workshop on YouTube. It's been posted on YouTube, so you can go back to this if you want to see something that I said or did or how to do something. So I think I got it to all, yes, got it. There's the YouTube video of this workshop. Okay, so the first is going to be a demonstration of these capabilities at the CONUS scale and showcasing the power of these tools for analyses that we're interested in. So I'm showing here two different study sites. The first study site is one that is in the Upper Colorado River Basin. This is the East River. And this work at the CONUS scale emerged from some of our science questions associated with the study sites, such as how are watersheds changing in response to snowpack conditions, and what is the role of watershed characteristics and functionality on that change, specifically related to nitrogen. So that work at that one scale has led to an examination of how water quality, specifically nitrogen and carbon, is changing across many U.S. watersheds. We can see that there are changes in the Upper Colorado, but that leads to the question of how does that relate to changes across other watersheds in the United States. The second picture here is the Russian River, and this is located in Northern California, and the Russian River has experienced multiple back-to-back fire years beginning in 2017. So each year we've had a fire within that watershed that has impacted the water quality in some way. So this concept of a watershed is important here because the watershed is an important integrator of changes that occur within the system upstream. So for example, if there's a fire in one part of the watershed, that fire and the signal of that fire will propagate down because of hydrological flow conditions. So what we're thinking about here is how do in-stream measurements reflect those coupled hydrobiogeochemical processes as well as changes? Are they indicators of that changes? And because water and solutes in nutrients, as we've heard about from all of our previous speakers today and yesterday, that there are reactions and there are microbes and there's a lot of biogeochemical change that occurs as water flows in rivers and in hyporheic zones and in sediments, and that all emerges at a particular point in the stream, which is that's where it ends up. And so we know that that's a reflection in some way of that hydrobiogeochemical coupling upstream. So the idea here being that if we look at these indicators across many different watersheds, we'll be able to understand something about either the watershed or the particular processes that are occurring in that watershed. So as I mentioned, this is part of our Watershed Function scientific focus area. If you're interested to learn more about that, our website is at the top there. And we're thinking specifically about how mountainous regions retain and release water, nutrients, carbon, and metals. So specifically for this demonstration today, the research questions I am focused on are how are trends in concentration, discharge, and export fluxes, loading is another term for that. How have they changed across the U.S., where we're using the watershed as a scaling construct and focusing on trends in those parameters? And how do those trends how are they changing, and how does the timing of fluxes change? For example, across the U.S., are mass fluxes of carbon and nitrogen to the oceans, is the timing of that changing? Is it occurring earlier or later in the water year? And then what is the relationship to other factors that variables that we have at the continental scale, such as atmospheric deposition or vegetation, remote sensing metrics that we can use to link water quality to remote sensing. So the methods that I'm using for this include a few R packages. So the R package is called data retrieval. Now this R package, this is one of the packages we're going to be using today in the hands-on demonstration that allows us to fetch the water quality data from the USGS. So that's it's like one line of code, and then you fetch the data, and then it in real time gives you that data back. So today we're going to be doing that for one particular station, but in this demonstration, you can imagine the computational needs if you wanted to do that for all stations, all of the USGS stations across the US. And then second, we used this other package called EGRET, E-G-R-E-T, which uses the weighted regressions on time, discharge, and season method. And so this method allows you to predict your concentration based on discharge using a series of statistical methods that link together concentration and discharge in what are called concentration discharge hysteresis curves. So these links to the R packages, I'm going to provide them in a link here in the chat window. You don't have to download any of these for our hands-on tutorial today. They're already implemented in the Jupyter notebook. So there we go. There's the links. And with the code that I'll show you today when we do the hands-on, you'll be able to reproduce this yourself either on your local machine or using the Jupyter notebook. If you use your local machine, then you will have to download these R packages. So in this demonstration here, I'm showing all of the different water quality parameters. There's a parameter ID. There's also a parameter name. So we're going to be focused on dissolved inorganic nitrogen, parameter 6631, there at the second line. So we're focused on that parameter today. And as I said, for this demonstration, you can't do it just using your Jupyter notebook because it's too big, but there are other R packages to help you accomplish analysis of big data sets. There's some functions that include using like parallel processing, MCL apply is one, using big mem packages. That way you can, like when you have 300 gigabytes of RAM that you need, you can use these packages to do this type of analysis. And so here is the example of what we get from that type of analysis. We get continental scale data and analysis, and we can really begin to zoom out, like what's happening at the large basin scales, and then zoom in for what's happening either at the station scale or at other hydrological unit scales. So here I'm showing multiple scales. For example, I'm highlighting the Pacific Northwest up at the top left because our host today, Pacific Northwest Lab, this is their basin, and we're looking at trends in nitrate concentration over the past 50 years. And so this is where the power really comes in of using some of these R packages. And so I won't dive into too many details of this, but if you do have other questions, feel free to reach out, because now I want to move into the hands-on part of our tutorial so we can have some fun during this next stage. Okay, So that's the demonstration of what you can do with these type of tools. Now we're going to start to do some of that. So we're going to look at one particular case on the Russian River, examining fire impacts. So for this next stage of the analysis, I'm going to start to switch over between screens here so that I can show you in Firefox how we're going to do this. We'll do this together, and then I'll send the link everyone's way so that you can follow along and do this as well. So I'm going to stop sharing really quickly and get that switched over. So first, while I get this switched over, I guess I'll check in with folks and see if there are questions before we move on.
I don't see anything in the chat right now or over in Discord. I think a lot of people are massively clicking on links that you've set out and maybe navigating over to your Firefox.
But I have to say,
okay,
this is awesome, Michelle, because we started the week, you know, really at the molecular scale, and now we've gone all the way up to the continent. So I love it.
I know, I'm looking, going, I know what I'm going to be doing this evening. It's just,
it's
just
really
incredible.
A lot of fun. It's especially when you see the hands-on tutorial today. You can change the station if you're interested in the Pacific Northwest stations. Yes. You can go take a look at your own. So I am sharing my screen, and I'm showing right now Firefox. Let me make sure that So in this first step, what we're doing, and let me make sure I get this binder link over to everyone right now. This is the binder link. This is how we're going to open up the Jupyter Notebook within Firefox. So as you can see, the binder, it's processing right now. It takes a moment to start. And okay, there we go. So it started for me. So I'll give everyone else a moment for it to start for them as well. Sometimes it can take a few minutes to start. And if it has started for you, then the next step is to click on the Jupyter Notebook. It's called rrwqfinal.ipymb. So you click on that, and that gets you into the notebook that's ready to run.
Yeah, great. Mine just finished loading, and I'm in. So thanks, Michelle.
Okay, good. So Tim got in, and everyone else's should start. The one part of this that I don't know what's going to happen is, since all, I don't know, 100 of us are attempting to do this at the same time, it's such a large scale that I don't know what will happen. So this will be a good test for that, I guess.
What could go wrong?
What could go wrong? But once you're in, and then once you click on the RRWQ Final IPYMB, then it takes you to the actual notebook that will run together. Okay, so someone else, Annette said they're in, so that's good. It's working for folks. Other people, okay, so I'm guessing we're having some smooth sailing thus far. So in order to run this notebook, all that you as the user have to do is click on Run. That's just this little button right here at the top. You just click Run, and that runs that particular section of code. So as you see, we're loading the libraries right now. I haven't updated the libraries in a while, so we get a few errors, but that's okay. Everything is still going to work for us today, so don't worry about those errors with the libraries. We're using multiple libraries today, including the data retrieval library, the egret library, leaflets, HTML widgets, ggplot2. You might have heard about these in other, I don't know, papers or tutorials, but specifically the ones that I called out earlier are data retrieval and egret, and that's how we're going to be fetching some of the data and doing the analysis. So good, I'm seeing more folks are in and able to run it, so that's great. Everything seems to be working. So what we're doing today within this Jupyter Notebook, we're exploring the impact of the North Bay fires on the Russian River. And if you don't know, in 2017, there were a series of pretty devastating wildfires that occurred overnight in the Russian River watershed. And it killed many people. Over 44 people were killed during this overnight fire event. And it impacted not only the rural regions, also urban. You can see a picture here where it swept through many neighborhoods. So this was a pretty devastating fire for this region. And after 2017, there have been fire events that have occurred. In 2018, there was another series of fires, and in 2019, there was the large Kincaid fire. So since 2017, we implemented with our collaborators, and our collaborators are folks at the U.S. Geological Survey. They include Jen Underwood, who is also on the call today as part of this workshop. So thanks for joining, Jen. And then there's others who are part of it as well. What we're doing is we're collecting water quality samples at multiple locations within the watershed, and these include tributaries and the main stem of the Russian River, and they allow us to supplement all of the U.S. Geological Survey data with our own data sets that are a bit more detailed. So we're collecting things like anions, cations, nutrients, mercury, for example. So we're really doing a large suite of analysis on those samples. And there's also within the notebook, there's links to some of the resources here. Specifically, if you look at the USGS water data website, that's where you can interactively look at a map and find stations that are available for your watershed. Okay, so as I mentioned, just after the 2017 fire, we implemented this sampling program, and you can read more about that sampling program at our news link. But this was specifically designed to capture the effects of the fire on the Sonoma County Water Quality Monitoring Program. The Russian River is used for drinking water through a series of riverbank filtration systems, so it's important to monitor the river for any changes that may happen. So we're going to use some functions that's going to be in this next step here to look at some of these data sets. We're going to automatically fetch the data from the USGS server, and then we're going to plot the data and map the data so we can look at essentially like across California, we're going to look at a lot of data. So this is the start of that. So in this next section here, these are called functions. Now, many of you who know you already know what a function is, but a function is a set of code that allows you to do something. You can think about it like maybe a mathematical function. You give it something and then it gives you something back. So we have two functions here, and we'll just click the run button. And then the functions are done. They don't really do anything right now until we call them later, but these functions, they've been run and they're going to sit and wait for us to provide some more information. So the next step is to actually gather the water quality data, and we're going to do this for one station. This is the Guerneville station. And so each site, each station from the USGS has a site ID. So I've already selected the site ID. This is the site ID for the Guerneville station. So the Guerneville station, it's not directly inside of a fire perimeter. However, if we go back to the concept of a watershed, this site is downstream of all of those fire zones. So all of the tributaries and streams, they eventually merge into the main stem of the Russian River, and then this site, the Guerneville site, this is the most downstream site. So we anticipate that this water will reflect the history of the travel path of any water particle. And so what we're going to do next, we're going to run section 4. This is where we're preparing our input values. So we have our site ID. This is the Guerneville station. We have the starting date for when's the earliest sample we want to collect. I have it quotes in blank quotes so that I can get the earliest value, whenever was the earliest data available. And then I have an end date. So this gets data up until July of 2018. But if you left this blank, it would get into all the data until the most recent end date. Here, this is the parameter code, and I initially thought we were going to look at dissolved inorganic nitrogen, but I'm wrong. This is the code for pH. So today we're going to be looking at pH. But you could change this parameter code. Let's say if you wanted to look at inorganic nitrogen or dissolved organic carbon or oxygen. So every water quality constituent has a parameter code, and you would input that parameter code there. Okay, so did I run that section? I will run it if I didn't already. There, that section's run. Then we're going to fetch the data. This is the section that will go grab the data, and specifically this is the line right here. This is the function get WQ daily, get water quality daily. And the inputs to this are the site ID, the parameter code, the start date, and the end date. And then, so it's gonna get that water quality, and I'm just gonna clean up the names of the column headers, so that way they have names that make sense, like date or month, decimal year, water year, the actual pH value. So I'll run this section of code. It should be pretty quick. Should take just, that was quick. Right over here on the side, if there's a little asterisk in the place of the 4, it means that it's running. So here we ran it, it looks good, and then we'll just take a quick look at the data. So if you use this line of code, click run, you can see what the data looks like. So we have our date column. On the left, we have pH, the value of pH in the second column, and then the other columns like the month, January being month 1, or the day, the decimal year, or the month sequence, or the water year. That helps you with doing, let's say if you wanted to do analysis by month or analysis by water year, then you can use these columns for that. And I specified here in this next section, what do all of the different components of the code mean. Okay, so now that we've collected our data, we can plot it and see what it looks like. So in this line of code, I'm going to plot the pH data by month. I'm interested in seeing how pH varies across the entire period of record of when it's binned by month. So what does the average January look like, or the average February? So in this line of code, we'll click run, and we'll get a box plot. And this is how pH varies across the water year. So January is 1, July is 7, And you can see there is a normal seasonal cycle associated with pH. So when there's any analysis of changes, let's say, you know, we go back to the question of how has fire impacted water quality? We have to know what are the baseline conditions, not only the average baseline conditions for the river, but also by month, because we know that there are seasonal changes. Even if we dig a little bit deeper and we look at daily time scales, we see that there are diurnal variations in pH. So there's a lot to consider when you're asking the question, how has, let's say, pH or nitrogen changed since this other period of time after something like a fire? So we have this as our baseline to work against. Next, what we're going to do, so we've taken a look at this figure. We see that there's some seasonal variability. We're going to start to think about the role of the fire. So we're going to classify the pH data using a simple pre-fire, post-fire classification. Pre-fire being, I will classify everything that's before September of 2017 as pre-fire, and then everything that's after September of 2017 as post-fire. So it's simply before and after. So that's this next step. We're classifying the pre and post-fire data. And then we can look at it. So now that I've classified it, I can look at how pH is different depending on that classification. So we run the next section of code, and then this gives us the next set of box plots. So it's similar to the box plot we just looked at, where we looked at pH as it varies across the year. But now we also have the classification. So we've split our data set into two categories, the pre-fire and post-fire. And so we see here in red is post-fire pH and blue is pre-fire. And so this is the distribution of the data for January, also now classified. So we can see for January, here's in general what the distribution is like pre-fire, and then here's the distribution post-fire. So we can see that the median has shifted upward, but pre-fire there's a larger interquartile range. So there's a lot of statistics to consider here when thinking about how has it changed post-fire. In February, this is when we see some interesting shifts. So this is the February data classified, pre-fire is blue, post-fire is red. Now we see a larger shift in the distribution of post-fire February data relative to pre-fire. Here we see the median is shifted more significantly upward than in January or some of the other months. But the distribution of all that prior data is much larger. And so this is a simple pre-post-fire classification, but one can imagine that to get more into the details here, we would need other factors. For example, atmospheric rivers and our drought conditions. That changes pH. So perhaps like the next step would be, okay, how does then dry year pH differ from wet year pH? And what are the differences in pH between those years? We know that the fires occurred during a relatively dry year. So the next step would be to further segregate the data into dry year and wet year conditions, and then really look at how pH varies across those different water year conditions. Okay, so then the next step, this is where we're going to get into some mapping. So this step here, we're going to do another data grab. This time, we're going to fetch data for the entire state of California. And we're going to grab a very limited range of dates because it's such a large data set. And It sometimes takes about 1 minute to fetch. So I'm going to start running this while I talk you through this. So it'll take about a minute to fetch all the data. Oh no, it was quick. Okay, for me it was quick. Hopefully for you it's quick too. And it's for the state of California. Specifically, we're getting pH from those specific start dates and end dates. And don't worry about the warning message. We can ignore that. So if we take a look at what this data looks like, so this is collecting data from multiple sources. It includes NWIS, but it also includes like seed end data comes here, other databases. I'm forgetting off the top of my head the name of those databases, but they're all included in this data set for the state of California. So you can see here, we can scroll through this data set to see more information about it. Okay, so in this next section, what we're going to do a little bit more refining of the data. We're going to do some averaging for the different stations. So I don't have too much time left today, so I'll just get to it, run that section, and then we'll further just look at what we just did. And then this is where we get into the mapping. So I'm in section 14. The mapping will allow us to see pH concentrations using colors as well as sizes, and it will be an interactive map. So once we run the leaflet map, and there we go. So this becomes an interactive map where we can zoom in onto the different locations where there's data, and we can click on these data sets to see where was this, what is the value, and yeah, so this is pretty fun to play with this to see how pH varies across the state of California using this cool data fetching technique. So I think with that I will stop and allow you all to ask me questions if anything wasn't clear. So thank you.
Thank you, Michelle. I was just going to say, if you had a few more minutes, we don't need to rush to wrap up. So we're going to our private session shortly. But but I appreciate you presenting this to us, and this is super interesting.
So there is one question, and I have to second that. It's really just fascinating. And seems really easy to use. There is a question, is this information only for the US, or can you gather data sources from other continents?
So this particular package and this particular set of code is only for the US, specifically because this is from the USGS. So this package has been built by the USGS for retrieving their data sets. So it is very specific to the US as of right now. I have not explored other databases or other R packages. For example, like if you wanted water quality for another country, there would have to be either a specific database that you go and you write your own line of code to go fetch it, or an R package maybe that somebody has developed for other water quality data sets. So yeah, it's variable around the world.
There was another question in here about, apparently I didn't see, but someplace on the map there's a river with a pH of 2. And
oh yes, I think I saw that as well. Let's zoom into that. Over here, I think, in Northern California, a value of 3 or a value of 5. Yeah, a lot of QA/QC needs to happen. So this data set, I've just done a quick fetch and a quick mapping. But if you wanted to, do more QA/QC and really dig in with, what's going on with this value or that value, that would need a little bit more work. Or even saying like, I'm going to cut off all data that seems weird. Anything above, for example, just 132 just seems weird. So you would have to remove those values. These values are suspicious. I agree.
Yeah.
That's
great.
That's really interesting. So I was going to ask, so Michelle, if we're playing around with this in your binder and we change parameters, because for example, thanks Nancy Marino, posted a link to all of the water quality codes for USGS. So if I wanted to change and look at, say you were saying inorganic nitrogen or something, I could change that code. How is that working in terms of me working on this binder? Is this like making a copy that I can play with, or am I messing you up if I change something, or how does that whole system work?
I don't know actually what would happen. That's a really good point, Tim. Yeah, I don't know if we all start playing with this one, what would happen, because I have these files locally. So I have them locally that it's pushing to a GitHub. So on the binder itself, the binder, like I have to put these files themselves on this binder. So I'm not really sure. Okay. I'm not really too worried because I can rebuild this stuff pretty easily.
Because it would be really fun if you don't mind us playing around with it and looking at it and trying some different things, but I didn't want to mess anything up. So.
Yeah. I think you're okay. It might not let you save it. Okay. I'm not sure. You can test it. Maybe if you make changes, I don't know if it would even let you save.
I think I saw something when I did change something, or I ran something, and then it flashed up briefly by the little window that says not trusted. It said something about file saved, but I don't know.
Okay.
It's auto saved up in the top, but anyway, yeah. I just saw a Discord question. Paula says, that was really cool. Which R package did you use to make the interactive map? And if that's something only for USGS data, would you know of anything else that could be internationally accessed? Paula's coming to us from the Netherlands.
Mm-hmm. Okay. So specifically for the map, I used Leaflet. So Leaflet is this package here that can be used to do any type of mapping for whatever data you have. So it's very broad. Now, specifically for this data that I mapped, it's only for the US, but I can imagine if you have your own water quality data set, you can do your own mapping with Leaflet.
Okay. Yeah, I don't, are there other questions out there? I'm sort of checking chat and our
looks good. Lots of
Discord.
Thanks and great job. So
yeah,
thanks everyone.
It was really wonderful presentation, and yeah, that's really cool. So that is interesting though. The interface through this, yeah. Whether we could clone one, save it as a different name. That's an interesting question. I guess it does say unsafe changes. So
yeah, I think you can definitely like copy these files, and then you can run the Jupyter Notebook on your own machine. It's the binder itself that I have set up to allow everyone to run R without having to install anything.